Today's study tip is to break up your study time a little bit. The optimal study time is about 20 to 25 minutes. Then you should take a 5-10 minute break and repeat. You could even make a list of rewards for yourself for each short break. You could get a snack, fold or put away laundry, go to the bathroom, take a phone call, play with your pet, short walk. It's funny how when you're studying, even things like folding and putting away laundry are rewards, right? But these things can break it up and help your brain take a break for a little bit before you go back to studying. Okay, so I want to point out that this topic is not one chapter. So the acid-base balance of blood is multiple chapters, but why we talk about it in this class is one, it's very important, and two, that nursing school at EVC personally requested that we cover a lot of acid-base balance. So because so many of our students from our courses go on to nursing school at Evergreen and other institutions, we wanted to make sure that we're covering this in depth, okay? So there's going to be a handout for this lecture as well, arterial blood gas analysis, and we're going to work more on this topic of acid-base balance in lab as well. Okay, so let's talk about the pH scale first. It goes from 1 to 14, and anything between 1 and a 7 is considered acidic. Anything from a 7 to 14 is considered basic, okay? And that 7 is neutral. Our blood is set point of 7.35 to 7.45, so slightly basic. 7 is true neutral. 7.35 to 45 of our blood is slightly basic. Okay, so anything below 7.35 then is considered blood acidosis. Anything above 7.45 is considered blood alkalosis. Okay, so our pH then is the negative log of the concentration of hydrogen ions. And we just said that negative 7.35 is acidosis. Below 7.0 can be fatal. At this condition, the CNS is depressed and you can enter coma and even death. 7.45 is alkalosis. So anything above that can be dangerous. Above 7.8 can be fatal. And this is because it overexcites the CNS, and so you can get muscle tetanus, extreme nervousness, convulsions, and death. So both are dangerous, and it's really this very narrow range, right? Lots of our set points have a larger range in which you can work under, but blood pH is one that requires very careful regulation, and movements outside of this narrow set point range are very dangerous. Okay. So what makes something an acid is that an acid is going to dissociate in water and donate hydrogen ion. And an increase in hydrogen ion equals a decrease in pH. So strong acids are going to completely dissociate. These are things like hydrochloric acid, HCl, whereas weak acids are going to only partially dissociate. So a weaker acid like carbonic acid that we'll come back to, H2CO3, is going to partially dissociate and donate some hydrogen ion then to our solution. Okay, so this is why weak acids are going to lower pH a little bit. Strong acids are going to completely dissociate and lower pH even more. Acids are made as a result of all different kinds of metabolism. So think about a lot of the ones you have in your body: fatty acids, lactic acid, carbonic acid. And so these metabolic products, being acids, become really important when we think about having to manage blood pH. Okay, so what makes something a strong or weak acid? Strong acids fully dissociate, like HCl. So here's full dissociation into hydrogen and chloride. And now you see there's more hydrogen ion than here available to lower the pH of the solution. Weak acids get partial dissociation, so you have some division into hydrogen ion and bicarbonate, but the rest remaining as carbonic acid. Okay, so bases also dissociate in solution, but they're going to accept hydrogen ion. So because they're accepting the hydrogen ion, you get a decrease in hydrogen ion in your solution then, and therefore an increase in pH. So these can be also strong or weak. Strong bases include things like sodium hydroxide. Weak bases include things like sodium bicarbonate. So how bases make pH more basic is going to be, let's think about this. So here's our beaker of water. Our pH is neutral. Why? Because we have balancing of these charges, okay? When you add NaOH, though, the NaOH is going to combine with some of these hydrogen ions, so removing them from the solution essentially, and now you have a decrease acidity, which is an increase in pH then. Okay. So buffers are incredibly important because they're used to prevent dramatic changes in hydrogen ion concentration when acids or bases are added, right? And we said that we have to maintain this very narrow range of blood pH, 7.35 to 7.45. So we need buffers in the blood to prevent these dramatic changes so we don't get convulsions or other dangerous effects of pH being too high or too low. And buffers are usually a combination of a weak acid and a weak base, because then you can deal with both sides, right? Buffers are going to be able to absorb and deal with hydrogen ion or basic solutions because you have this combination of acid and base in the buffer. So if your solution becomes acidic, the weak base is used to counteract the problem, and vice versa. If the solution becomes too basic, the weak acid is used to counteract the problem. Okay. So you did this in lab, where you have a buffer solution and an unbuffered solution like water. You saw that there were drastic changes in pH when you added sodium hydroxide or hydrochloric acid. In a solution with a buffer, though, you can add base and you can add acid, and you don't see drastic changes in pH. Okay. So, incredibly important, you're going to have to memorize and be able to use our acid-base balance equation. Okay. So the bicarbonate buffer system is the most common. We have carbonic acid, H2CO3, as our weak acid. And this is easily made by changing the respiratory rate. Okay. The bicarbonate ion is our weak base, HCO3 minus. And a high concentration is kept in the blood to be able to counteract any acidic changes that happen in the blood. Okay, so what's our equation we got to know about? It's going to be CO2 plus water, carbonic acid, hydrogen ion plus bicarbonate. Okay. We also do have a protein buffer system, which is more plentiful but is not used as often. Okay, and this is actually used in intracellular fluid and plasma, where you have COOH end of protein is a weak acid, and this is because it donates hydrogen ion to become COO minus. The NH2 end of the protein is a weak base, and this accepts hydrogen ion to become NH3 plus. Hemoglobin in red blood cells also can donate hydrogen ion like an acid when it binds to CO2 in the tissues, and accept hydrogen ion like a base when it becomes H hemoglobin or reduced hemoglobin. And then there's a phosphate buffering system. So that first one to know is going to be the most important, but these are all buffering systems because they have a combination of a weak acid and a weak base. The phosphate buffering system is used in urine and intracellular fluid. And so NaH2PO4 is your weak acid. Na2HPO4 is a weak base. So again, buffering ability then of multiple systems. Okay. So we put this right next to respiration because respiratory system is a main way in which you control pH. Why? Because it's going to be a pretty fast way in which to deal with carbon dioxide, which is acidic. Okay. So you can alter the pH by changing the concentration of carbon dioxide. When we consider this bicarbonate buffering equation, let's think about this. If you get lots of CO2 blown out of the lungs, the shift left will do what? And what will happen to pH? This will decrease. We're getting a shift left. Okay. You'll get a decrease in hydrogen ion, and therefore pH will increase. This is respiratory alkalosis, an increase in pH in your blood, alkaline, caused by a change in respiration. In the opposite direction, if little CO2 is blown out of the lungs, you get a shift towards the right. Hydrogen ion then will increase and pH will decrease. This is respiratory acidosis. Okay. Interestingly enough, though, if a person is locked in a completely sealed room, they will first die of carbon dioxide poisoning before they die of oxygen deprivation. So your body responds more strongly to carbon dioxide than it does to lack of oxygen. So certainly we need oxygen, it's important, but your body monitors and regulates carbon dioxide more carefully because of the catastrophic effects of carbon dioxide poisoning. Okay. So the other place in which we're going to regulate acid-base balance, besides respiration, is going to be in the renal system. So again, how important the kidneys are, right? We talked about renal physiology recently, where it's going to create urine, of course, but then the renal system is also going to regulate ion balance and now acid-base balance. Okay, so let's look at our blood, comparing to the lumen here and the filtrate that's happening inside. We're going to see mechanisms here in order to adjust pH by regulating things across the wall between the blood and the lumen. Okay. All right. So first, we can have bicarbonate reabsorption. You can have hydrogen ion secretion, but note that those are the only two options. So you can reabsorb bicarbonate, but not secrete it. You can secrete hydrogen ion, but not reabsorb it. Okay, so the mechanism here is that you can reabsorb bicarbonate by having this buffering system we just talked about here in the lumen wall. Okay, so here is carbon dioxide and water. If that's present in high amounts then in the urine, it's going to become carbonic acid then in the wall here, and that can then dissociate and bring bicarbonate back into the blood, so reabsorb the bicarbonate. Okay. For the hydrogen ion side of things, you can pump out hydrogen ion into the lumen. Okay. But pumping is really happening here with this sodium hydrogen pump here. But again, we're seeing the same equation happening on the other side, right? Carbonic acid and then dissociating into water and carbon dioxide here too. Okay. So main point here is that you're going to be able to secrete hydrogen ion and you're going to be able to reabsorb bicarbonate. Okay, so how do we do this? For hydrogen ion secretion, if you have an increase then in hydrogen ion in the blood or a decrease in pH in the blood, same thing, then you're going to have increased hydrogen ion secretion into the renal tubules. And the opposite is true. If it's too low in the blood, you're going to have decreased secretion. For bicarbonate reabsorption, if you have increased hydrogen ion in the blood, you're going to have increased CO3 reabsorption in the renal tubules. And the opposite is also true. Okay. NH3 can bind hydrogen ion and can be released in urine if that ion is too high. All right, so let's go through some examples here. If there is metabolic acidosis, so when we talked about respiratory acidosis, alkalosis, if we have metabolic acidosis, then what would compensation be? The way to compensate for metabolic acidosis then is going to be respiratory alkalosis. So this is indicating that because of metabolism, you're getting too much acid, pH is dropping. You can compensate for that by respiratory alkalosis. How would you do that? You would increase your breathing rate to dump more CO2 from your breath. So hyperventilate is going to allow respiratory alkalosis to compensate for metabolic acidosis. What will this do to PCO2 levels? It'll decrease them. For hydrogen ion in the blood, decrease because you've blown off that CO2 and that will decrease hydrogen ion in the blood. The result is that your pH then will increase from your metabolic acidosis state back to hopefully normal. All right, different example. If there's respiratory acidosis, think about what the compensation might be. Would it be respiratory or metabolic? Would it be acidosis or alkalosis? Okay. Respiratory acidosis is going to be balanced, compensated for by metabolic alkalosis. Okay. So to do this, our patient is going to need to do what to H plus secretion? They're going to have to increase it, right? Trying to get rid of the acid here in the blood. And they're going to increase CO3 reabsorption because if you're going to have more CO3 reabsorbed, now you're going to be able to balance the hydrogen ion that's in your blood. Okay. This will eventually decrease the hydrogen ion present in the blood, resulting in a pH increase. Okay. Now a case study. Let's imagine that you went to Japan before the pandemic, okay? And you climbed to the top of Mount Fuji. Mount Fuji is 12,388 feet, and along the way there were small huts selling snacks, water, and small cans of pressurized oxygen. So why would someone be selling oxygen for an outdoor activity? Let's think about that first. Okay. And then, as you approached the top of the mountain, there was a middle-aged Japanese man sitting along the pathway, breathing rapidly. He was obviously out of breath and looked very ill. What could be going on with the acid-base imbalance in this man and the cause of it? Okay, so note breathing rapidly. What might that indicate? And what might that indicate then about his acid-base state? So I want you to describe the compensation he would have and the physiological significance there too. Okay. So you're going to work on your arterial blood gas handout. We'll talk about this more in lab. But you got to start with thinking about what our normal conditions are going to be. Okay. So to get you started on this, here's our normal conditions. Our pH of the blood normally is going to be 7.35 to 7.45. At this level, the PCO2 in millimeters of mercury is going to be 35 to 45. Bicarbonate levels are going to be 22 to 26. So when you move into an acidic state, 7.34 or lower, you get increased amount of PCO2 and a decreased amount of bicarbonate. Okay. When you enter a basic state, 7.46, you see decreased PCO2 and increased bicarbonate. Okay. And so this is all about balance. It's another homeostatic mechanism we're learning, all about balancing pH. With your systems able to balance pH and manage pH are going to be the lungs and the kidneys. So whenever you think about this, think carefully about where is the acid coming from? Is that going to be then alkalosis, which is raising pH, or acidosis, decreasing pH, and then how your body might compensate? Okay, and so we'll work on this some more in lab and get a really good understanding then of acid base.